Hello dear students, this is the second video of Light Reactions lecture series. This is in continuation with the first video, so please watch the previous video first, then only you will get a connection. So let's see the non-cyclic electron transport and photophosphorylation in detail. Well, let's study the non-cyclic electron transport in detail. Here, this is the representation of non-cyclic electron transport and the scheme. Here you can see two pigment systems are there, pigment system 1 and pigment system 2, and also you already studied P700 is the reaction center here and here it is P680. Here you can see PS1 is accepting light and PS2 is also excited by light and electrons are ejecting. These processes are here and also water is getting broken up in the photolysis of water. So all these processes are the parts of non-cyclic electron transport, and all these processes are taking simultaneously inside thylakoid membrane. But for better understanding, I am explaining this whole process as three different phases. The first phase is movement of electron from PS1 to NADP+. The first phase is movement of electron from PS1 to NADP+. Second step, movement of electron from PS2 to PS1. Second phase is movement of electron from PS2 to PS1. Third phase is photolysis of water. So let's learn what happens in these three phases. The process is initiated here by absorption of 2 photons by P700. So we know when it absorbs light energy in the form of photons, it is excited to higher energy level P700. So the excited state, it is represented by this sign mark. There is a small mistake. Here you should not write 2 electrons here. Electrons are emitted or ejected from this particular state. So that is why put or write these 2 electrons here. So please correct it. So now electrons are emitted from this excited chlorophyll A molecule and it should be accepted by some electron carriers. So which compound act as the electron carrier or electron acceptor, the primary acceptor? So there are different views regarding this primary acceptor compound. According to one view, electrons are trapped by the membrane bound iron sulfur proteins. According to some others, there are some other molecules also like A0, A1 before this iron sulfur proteins. So in order to avoid all these confusions, I would like to represent it as ferredoxin reducing substance. So there is another view of representing this as ferredoxin reducing substance, FRS. So I think this is better. You should write like this. So this is to avoid confusion. So just write ferredoxin reducing substance. Ferredoxin reducing substance means it can be whatever it is, A0, A1 or membrane bound iron sulfur protein. So you just write it as FRS, ferredoxin reducing substance. So this part absorbs or it accepts electron from P700 and it donates this electron to nearby lying ferredoxin. That is why the name ferredoxin reducing substance. So this compound reduces ferredoxin. So that is why we are representing the electron carriers, whatever it is, membrane bound iron sulfur proteins A0, A1, we are representing by that simple term ferredoxin reducing substance. So P700 releases electrons and they are accepted by FRS, and from FRS it is going to ferredoxin, and from ferredoxin what happens to these electrons? These 2 electrons are passed on to NADP plus through an intermediate compound ferredoxin NADP plus reductase. Now these 2 electrons are passed on to NADP plus. Now NADP plus is partially reduced, not fully, because for the full reduction it requires 2H plus also. Now 2H plus are not arrived into the scene. So we are just pausing the reaction here and we are waiting for the arrival of 2H plus. Okay. So let's see what happens to PS2 at the same time. Here we know P680 is the reaction center in pigment system 2, and it is also absorbing light in the form of photons and excited to high energy level. So this is the P680 molecule which is excited, which is represented by the star mark. And here also the same mistake is repeated. So you just notice this. Please avoid this 2 electron. Here you should not write 2 electrons here. Instead of that you write 2 electrons here. Okay. So 2 electrons are now ejected from this P680 which is the excited condition, and these electrons are primarily accepted by phyophytin. Phyophytin is the compound which is taking electrons from this excited P680, and from phyophytin these electrons are passing a downhill way. It is passing through a downhill way towards PS1. That is the important thing. So from phyophytin electrons are moving through a downhill pathway towards pigment system 1 through a series of electron carriers. So you should remember the names of these series of electron carriers. So it's moving through a series of electron carriers. This is a downhill movement. That means an energy release is taking place, and here the electron after losing the extra energy, the electron not coming back, it is coming to pigment system 1. Okay, so these electrons from phyophytin, now the electron is with high energy. So in this moment the electron loses its energy and it is returning, it is coming to PS1, P700 to fill the hole. We know after the ejection of these 2 electrons there is a hole or electron deficiency existing in pigment system 1. So these electrons are utilized to fill the hole or electron deficiency in PS1. Okay. Here the series of electron carriers consists of plastoquinone, cytochrome B6F complex and plastocyanin. Here the cytochrome B6F complex, it is termed as risky center. This is another name for the cytochrome B6F complex. So you should remember the term. So here in this particular point, in between plastoquinone and cytochrome B6F complex, there occur, when electron flows through this particular point, there occur a sufficient amount of free energy change or enough free energy change which allows the photophosphorylation to occur, or which allows the phosphorylation of one molecule of ADP with inorganic phosphate to form ATP. So one molecule of ATP is produced during this electron transport from PS2 to PS1. So this is known as photophosphorylation. So you just remember the position where ATP is formed, and we should study it later. We will discuss it later, the exact mechanism. So you just now you just remember the position where ATP is synthesized. It is in between PS2 and PS1, exactly in between plastoquinone and cytochrome B6F complex. So that's all. And now the electrons now reached PS1 and it fills the hole of PS1. Okay, but still now the hole in P700 of PS1 is it has been filled, but at the same time a hole is still existing in P680. So who will help PS2 to fill the hole? Here comes water. So now we are moving on to the third phase, that is photolysis of water. Here water acts as the electron and hydrogen donor, because water is splitting up to produce 2H plus, 2 electron and half O2, and these 2 electrons are supplied to P680 to fill the hole of P680, and the 2H plus produced by this splitting up of water is utilized here for the reduction of NADPH. Now by the arrival of 2H plus from photolysis, NADP plus is now fully reduced to form NADPH. So now ATP is synthesized here and NADPH is also synthesized here. And as a result of photolysis, oxygen molecule is produced. Oxygen molecule is also produced and this is released as a byproduct of photosynthesis here. So all these three processes occur simultaneously and they constitute the non-cyclic electron transport. So this is the representation of non-cyclic electron transport. So we learned it as three phases. Okay, but these processes occur simultaneously. And here why the name non-cyclic? Here electrons ejected from one position is never returned back to original position. They are used up somewhere. Okay. Here we know electron ejected from PS1, it is utilized to reduce NADP+. They are not returning back to PS1, never returning back to PS1. And at the same time, electrons emitted from PS2, they are never returning back to PS1, but they are used up somewhere where it is used to fill the hole of PS1. Okay, so that is why the name non-cyclic electron transport. And non-cyclic electron transport possesses an important process, photolysis. Okay, so it non-cyclic electron transport possesses one important process, that is photolysis of water, in which water is splitting up by oxygen evolving complex of pigment system 2. So this is very important. Pigment system 2 is directly involving in this photolysis and oxygen evolving complex is taking part here to release protons, electrons and molecular oxygen, and the protons released from this photolysis is utilized to reduce NADP plus to NADPH, and electrons are used to fill the hole in PS2, and molecular oxygen is also released as a byproduct. Okay, let's learn some more points about photolysis of water. How can we define photolysis of water? It is the light-induced splitting up of water by which water is getting oxidized. Light-induced splitting up of water, as a result of which water is getting oxidized to release oxygen. So it is also known as photooxidation of water. So photolysis or photooxidation of water. Well, have a look what happens when water splits up. Here four water molecules splits up to produce 4H plus and 4OH minus. These 4H plus are utilized for the NADPH formation. So 4H plus are donated to NADP plus and NADPH is forming. So what happens to remaining 4OH minus? These 4OH minus are rearranged to form 4 electrons, 1 molecule of oxygen and 2 water molecules. So these 4 electrons are given to pigment system 2 to fill the hole of P680, and the oxygen is released out as a byproduct and 2 water molecules are regenerated. So here half of the water molecules are regenerated. Out of the 4, 2 water molecules are regenerated. So here you have to remember the modified equation of photosynthesis that you have studied earlier. Here we found that 12 water molecules are given here, out of which 6 water molecules are utilized and 6 water molecules are regenerated. So I hope now you understood why this 6 water molecules are regenerated. So this is clear from here. Here excess water molecules are needed for the smooth conduct of photolysis. And one more important thing regarding photolysis. Photolysis is associated with oxygen evolving complex in pigment system 2, and they are catalyzed by the presence of manganese and chloride ions. So the presence of manganese and chloride ions are essential for the smooth conduct of photolysis of water. So remember these points. The presence of manganese and chloride ions are very much needed for photolysis of water, and in addition to that, an unknown strong oxidant Z, it is also supposed to be involving in photolysis. Okay. Now I would like to summarize the non-cyclic electron transport that we have studied as following points. So this is a very simple representation of non-cyclic electron transport. Here we can see both pigment systems are involving in non-cyclic electron transport. That is one important feature of non-cyclic electron transport. Here both PS1 and PS2, here PS1 and here this is PS2. So both pigment systems are involving in non-cyclic electron transport. That is the first important point. And the second one, here photolysis of water is taking place. Water is splitting up here. In non-cyclic electron transport, we can see the splitting up of water. Here water is splitting up, so that is photolysis of water. And here water is actually splitting up for donating these protons, primarily for donating this proton. Actually, what is the role of water in light reaction? It acts as the proton donor. Water molecule acts as the proton donor for NADPH synthesis. So remember this. What is the function of water in light reaction? Water acts as the proton donor. That is the primary function of water, and for that function, photolysis of water is taking place. And molecular oxygen is also released as a byproduct during this photolysis of water, and this is also highly significant. Actually, in the process of photosynthesis, molecular oxygen is just a byproduct, but this particular process is very much significant for the existence of life on Earth. And we found, along with this non-cyclic electron transport, both ATP and NADPH are produced. ATP produced here in between PS2 and PS1, and NADPH is produced here. So both assimilatory powers ATP and NADPH are produced in non-cyclic electron transport. Then why the name non-cyclic, that we understood earlier. Path of electron is non-cyclic. Ejected electron, that is why ejected electrons are never returning back to the original position but used up somewhere. That is why the name non-cyclic. And one more important thing, non-cyclic electron transport is also known as Z scheme. Why Z scheme? This electron transport takes the shape of Z. That is why the name Z scheme. And here we found ATP synthesized here, and ATP synthesis in light reaction is termed as photophosphorylation, and here this particular photophosphorylation is termed as non-cyclic photophosphorylation, because this ATP synthesis is accompanying with the non-cyclic electron transport. That is why it is non-cyclic photophosphorylation. Okay. I hope all of you understood the process of non-cyclic electron transport and photophosphorylation well through this video lecture. So let's see in the next video with cyclic electron transport and photophosphorylation. Okay, bye.